Thanks for the kind introduction. Today I'm going to talk to you about the use of process analytical tools in the development of continuous flow processes. This is going to cover from an R&D concept through to industrial scale implementation, using case studies that will highlight the type of development work that you do at each stage. So Chemtrix is a continuous flow reactor manufacturer, and we focus on the development of scalable flow processes. And we work together with partners ranging from universities to industrial manufacturers and system integrators to deliver total solutions. So how do we go about designing a process that we believe will go from lab through to manufacturing using continuous flow processing? We start with theoretical evaluation. This is critical to define the process targets and really get to grips with the actual problem that you're trying to solve by developing a flow process. Within this phase, we also look for theoretical opportunities for process improvement, and together with the end user, discuss those opportunities that are viable or not viable for a given business case. We then move on to a basic feasibility study. This will involve lab scale demonstrations, and the goal is to deliver proof of concept on the theoretical evaluation. We look to, at the outcome of this, outline milestones for process optimization, which will take place in a subsequent step. And this is typically a go or a no-go stage for a flow process development. If it's a go, we'll then go into parameter optimization, in which we're looking into detailed interrogation of the process, understanding critical process parameters, and evaluating suitable hardware to reach the scale that the actual process needs to be. So production can be a range of scales depending on the industry you're in. Pharma might need a few tens of kilos of material. Fine chemicals, you're looking at a couple of 100 metric tons a month. So at this stage, we're looking at the viability of the process towards the hardware available and the business case associated with it. Again, should this go through into the next stage, we'd look to realise that into a production plant. So this would be with an industrial partner, and in this phase you're going to design the plant, build and test it, and importantly, commission it. For those of you that are new to continuous flow reactors, I'll just give you a brief overview of how we're using these devices, and this will hopefully give you an idea of how you can use them in R&D or your production environment. So I'm focusing on the use of static flow reactors, and this means that we work with solutions typically. And what you can see in the schematic is we have two solutions. We pump them from separate pumps, and we bring them to a mix point that's thermally regulated, and we collect the reaction mixture at the outlet. So if we compare this to a batch process, we're typically designing and engineering that flow setup to suit the process that you're wanting to perform, in comparison to a batch process where you'll often change your process so that you can control it within the batch vessel that you have. If we look at those elements in a little more detail, at the heart of a good flow system is a stable dosing system. So the pump is critical. You've got to choose this based on the flow rate ranges, the pressure, and the materials that you're wanting to pump. If you have a poor performing pump, you're going to have an unstable flow process. No matter how good your reactor is, you will have deviations in your process quality. Next comes the flow reactor. Now the reactor itself needs to perform a series of functions. So you've got your thermal regulation of your feed materials, mixing, and thermal regulation of the reaction mixture whilst you're giving it enough time to react. You also need to consider that you can operate at the intended pressures, temperatures, and that the material of construction is suitable for the reagents and intermediates byproducts that might be generated within the reactor. Compared to batch, flow processes typically employ very short reaction times of the order of seconds through to minutes. Whilst this is a benefit from a throughput and a high performance perspective, it can be a challenge from ensuring that your product quality is consistently produced. So one way of doing this is to continually monitor what's happening in your process, and that can be through the use of PAT tools. You can see a couple here in the photographs. We've got a FTIR, which is a flow cell at the outlet of the reactor, or a Waters Patrol system that's taking sample aliquots and analysing them by UPLC. The choice of PAT, as we'll look at through this presentation, is dependent on the product that you're making and the process sensitivity that you're wanting to guarantee. Ensuring that you generate a stable product quality is part of the process development. You also need to look at how you're going to isolate that material once it's generated. Are you going to put the product into a batch isolation, or are you going to look to do that also continuously? So here you can see an example of a liquid-liquid separator from Zyput, and here this would be a continuous liquid-liquid extraction following on from the flow reactor. So again, reaction is only part of developing a continuous flow process. You also need to look at the downstream requirements. Once you've developed your flow process and shown it to be robust, you're going to then want to go into production. So here you can see an example of a modular skid-based setup incorporating reagent feeds, the reactor, thermal control in a suitable environment for manufacturing. That might be, in this case, a walk-in hood. It could also be a production plant. So you need to know upfront, is this going to be in an explosion-proof environment or not, so that you can choose appropriate hardware when you're actually doing your development work. So if we look at how a flow process differs to batch, in your batch reaction, your process is evolving over time. So to do different experiments, you would essentially restart your batch reaction with different reagents, different equivalents, different conditions, and monitor that process over a period of time. In a flow reactor, time is a function of distance within the reaction channel, as you can see on the right hand side. So as a result, you're optimizing processes differently. In a flow process, by changing flow rate ratios, you can instantly change your stoichiometries. By changing overall flow rate volume, you can be changing your reaction time, and on the fly you can change your temperature. So what you're able to do is much more quickly, in a data-rich environment, generate lots of different reaction conditions. This can, however, be a challenge because you're really quickly generating different samples, and this can give you a backlog if you're looking at offline analysis. So typically, the most efficient way to design and develop a flow process is to use process analytical tools so that you're able to be constantly monitoring the outlet of that flow reactor and minimise the amount of reagent you use to develop your process. I just mentioned that a continuous flow reactor can generate a large amount of data in a short amount of time. So it's disadvantageous if you're collecting samples, analysing them by HPLC or GC, and finding out, was the process any good, yes or no, 20-25 minutes earlier. So by having an online tool, you can speed up the data collection, and you can also learn a lot more about your process. For offline tools, you generally have to quench your reaction mixture to make sure it's stable. You might have to extract it so that it's in a suitable medium for the analytical tool itself, and this can lead to a loss of information. So not only are you losing time, but you might miss transient intermediates, unstable intermediates. You may racemize your product. So a lot can change if you're actually stabilising your sample ready for later analysis. In addition, we look at using PAT as a method of process control. So in real time, you're able to look at your process and say it's in a state of control. We can now collect that material. It's of sufficient quality and it's operating as a process as we intended. You can also use PAT as an alarm system. So are you having any drift in your process? Are you now starting to operate outside of your critical process parameters? And should that material now be diverted to a hold tank for further assessment whilst you bring your process back online? So at an early stage, you're using PAT to inform you of how to develop your process and how to better optimize it. And at a later stage, when you're looking to manufacture, you're using it as a means of controlling your process. So there's a lot of information that you can access through PAT, and it ranges in the early stages to conversion and kinetic profiling, understanding of your reaction process, your reaction mechanism, any intermediates or byproducts that can be forming. It can also give you insight into your reaction performance. This could be flow distribution, mixing, residence time distribution within your device. And there are tools coming into the market now for reaction calorimetry. So what you have to be mindful of when you're developing your flow process is what's your technical need at a given stage versus the business need. So you might need a tool that can give you deep insight into your process when you're developing it. But in order to run a stable, well-performing process, once you understand your design space, you may need simple tools like pressure, temperature measurement, and mass flow, for example. So it's important that you understand what you need at what point, and then if you build this in at a feasibility stage, you can prevent the development of an unviable process when you consider taking it through to manufacturing. So if we look at the use of PAT under process control, what we're looking for is monitoring a change in the process. And here you can see at startup we go from having no flow to flow now coming up to the set point, and we're measuring over a period of time no deviation from the set point. This green period can be determined as a state of control. So you're operating within your design space, you're operating at your set point, therefore your product quality is of a high enough degree for you to start collecting it. What happens when we have a disturbance? Well, you can see in the centre here we've got a drop in flow and it's quickly recovered. During this disturbance we stay within the design space, and this means the product quality is unaffected, and you can say we're still in a state of control and the material is continually collected. Later you can see we have an out of control event. So the flow now drops, we go out of the design space. So this would typically be triggering an alarm, and it takes a significant period of time to recover, never fully recovering to the design space. So this period, the material should be diverted to waste because you can't guarantee the product quality, and this could go in a hold tank waiting for further assessment, or it could be disposed of. So what you can see is that if you were not analysing every second, for example, you would miss this period of disturbance, and you would be continually collecting your material. As a result, the material in the end of the process run contaminates your clean, high quality material from the start of the run. So by relying on end of point testing only, you can miss these disturbances and you reduce the quality of your overall material collected. In addition to a process disturbance, PAT can also give you insight into process drift. So what you can see here is for the bulk of the schematic we stay in a state of control and the material quality will be of the predetermined quality. However, clearly something's going on in this process that's leading to the gradual increase in the signal. If we couple PAT together with advanced process control and model predictive control, what you can start to do is understand the root cause of that change, and maybe you start to do preventative actions much sooner than waiting for your process to go out of the design space. So this can save you a lot of time, it can save you diverting material to waste, because you can much earlier correct the root cause of this change. So drifting is an indication of a problem. The question is, what is the problem and how much impact does it have on your process, and will it lead to a loss of quality or a need to have to shut down your process for cleaning, for repair, etc. So there's many different PAT tools that you can be using. What we're going to discuss is that these can range from anything from temperature, pressure, flow, conductivity, through to FTIR, Raman, UV, mass spec. We're seeing a lot of online NMR tools being used also. I hope what you've been able to see so far is the differences between using PAT tool for optimization and for control purposes. And what I'd like to give you now are some examples of how we've used PAT in process development. Firstly, to gain process understanding, and then in the translation into larger kilogram and ton scale production. So the first piece of chemistry I want to show you today is the preparation of an organic azide. And this is a process that we initially investigated together with an industrial partner, but now on a simpler molecule that I can show you, we're able to do demonstrations generally at flow conferences together with METTLER to show you how quickly you can optimize a process and where PAT brings you a benefit. So from a synthetic perspective, performing synthesis of azides under continuous flow has a number of benefits. Safety is critical. So the process has no headspace in a flow reactor, and this means you've got no risk of hydrozoic acid buildup. So this increases the safety profile of performing these reactions at scale. We can also more easily access novel operating windows, so we're much more easily able to achieve high temperatures, high pressures in a flow reactor, and this means you can come out of the normal operating window for the azide production and actually accelerate it into tens of seconds by increasing the temperature. Conversely, because we use really short reaction times, we're not going to see thermal decomposition of the azide. So what we can do when we're monitoring this process is firstly look at the consumption of the bromide, the conversion into the organic azide, and also the thermal stability of that organic azide over a duration. So by having eyes on the process and using FTIR in this situation, we're able to look at the consumption of the starting materials and the formation of the product alongside any side products or decomposition routes. So to do this, we're using these small glass reactors that you can see on the right hand side, and in these we're able to preheat the feed solutions, mix, give sufficient reaction time, and if necessary quench, but in this case with the online analytics we don't need to do that. So what you can see here is a setup of the small reactor. We've got a temperature controller, pumps, and our reactor holder, and then you can see the flow cell at the outlet of the reactor. So initially we're pumping through the raw materials, and you can see on the right hand side that we have our spectra. Key feature for us in this reaction is the sodium azide signal. You can see it's away from everything else, and we've got a really nice sharp peak here. So we start by monitoring the raw material, and the consumption of this peak will give us an indication that the reaction is taking place. In the 3D plot here over time, you can see that we've got a stable signal on that azide peak compared to the water, which is the solvent. This is important because you want any change to be attributed to your process and not due to drift in your sensing or change in your reaction mixture stability. And what you can see is as soon as we turn on the thermal controller and we increase the temperature, you can see on the left hand edge that we start to decrease that sodium azide signal. And at the same time you can see we get an increase in a different area of the spectra. And in this case we're just over 2100, and this is the signal for the organic azide. So at the same time as monitoring the loss of the sodium azide starting material, we can also monitor the appearance of the organic azide product. On the graph you can see that in the flow reactor we're getting very little reaction under conventional thermal conditions of up to 50-60 degrees. However, this process is very strongly thermally activated, and you can see by working in a novel operating window that you conventionally wouldn't use in batch, we can accelerate this process to be giving you quantitative conversion of the alkyl bromide over to the organic azide in 30 seconds. You can also see how quickly we're able to screen different raw materials, looking at the impact on production cost, and you can see that the chloride, as expected, is much less reactive, but given the fact it's significantly cheaper, in this case you may consider to extend your reaction time and work with a cheaper raw material. So what we can get by working in this way is a really early indication of viability of manufacture, insight into key influences on the product and byproduct formation, and really quick data generation on small quantities of material. Once you've done your detailed interrogation of your process at the milligram stage, you're obviously going to want to increase the amount of material that you can produce. So a flow reactor produces more material by running it for longer or by increasing the volume of the reactor. And what you can see in this example is we looked at the aminothiazole core motif that's often found in pharmaceutically relevant materials, and we looked at the development of a process at lab scale, scaling through into kilogram material production, and we used the FTIR in order to determine firstly the optimal conditions, but secondly long-term stability for the operation of the reactor. Here you can see a schematic of the flow reactor for the development work. So we've got the ketone, the thiourea, coming in from individual feeds. preheating, mixing, specific reaction time, and prior to using the inline flow cell, we were quenching this reaction mixture with DMA in NMP in order to obtain a stable aminothiazole product for offline analysis by HPLC. What we were able to do by coupling with the FTIR is we were really quickly able to screen through looking at stoichiometry influences, temperature from 25 to 195 degrees, and the stability of the process to long-term operation. And when you're using a small flow reactor like this, one of the benefits is in a couple of hours you can have done several hundred reactor volumes to demonstrate that you have a stable process. So what you can see here is the spectra on the right, and this is a summation of the data that we collected over a range of temperatures. And you can see several areas of interest where we're getting change over the processing conditions that we've applied. So if we take key areas of the spectra that I just showed you that are unique to the starting material and product, we can plot the intensity of those signals against one another and monitor what happens to them as we change temperature. What you can see in the graph on the left is we're gradually increasing the temperature by 10 degrees, and each time we do that we get a loss of starting material and we get a corresponding increase of product. And by doing this, within 40 minutes we're able to go from a very low conversion of the ketone to quantitative conversion and formation of the aminothiazole. We're then able to take this information through into a larger scale reactor working at about 127 grams an hour from a few milligrams per hour, and we can optimize that process, generating kilos of material per shift. In the previous examples I've shown you the use of FTIR with stepwise condition changing, but in 2011 we actually demonstrated that you could extract kinetic profiles from flow reactors using Raman, operating under a non-steady state environment. So what we're able to do is constantly ramp the flow rate, giving access to different reaction times within the device, and plot this against a model. We're able to extract the reaction order and the rate of reaction, comparing it with conventional models. And by doing this we found that it was 5 times faster than doing this in batch or working with a stepwise profile. So if you want to know more about this, there's a JAX paper that can give you all the maths behind how you do this process extraction. FTIR, we were able to optimize the process conditions to have a reduced amount of anhydride and also give each stage the ideal reaction time and temperature. So what you can see here when we're telescoping the reaction steps, after the first reactor we have a flow cell that's confirming whether or not we've consumed all of the anhydride. We then have the third pump coming in, bringing in the acid source. In the second reactor you get the dehydration, and again monitoring at the outlet of the reactor with another unit we're able to confirm that the dehydration is completed. It's very useful when you're looking at starting up a flow reactor that you're able to have inline tools like this, because you can first start up the first reactor, equilibrate that to waste, and then when you've got that process under a state of control, bring in the pump for the third feed and start up your second reaction. So this can be time and material saving. Here you can see a close-up of the 2 reactors. So in the back we've got the cooler reactor, and at the front we've got the higher temperature, and you can see the older model of the RATIONALE and the product mixture being collected. And what we're able to do here is then get a greater than 99% yield on this process, operating at the kilo level for the generation of the API intermediate. In addition to spectroscopy, people are also looking at other techniques like mass spectrometry. So Graham Cooks at Purdue University has been working on the development of a fast reaction screening tool based on DESI mass spec. And in this we're able to use the flow reactor to rapidly generate a large number of samples and then use mass spec to interrogate low-level impurities, for example, in the APIs that are produced. So this again is an early indication of viability of manufacture and allows you to make the right choices over raw materials, solvents and process conditions. A recent example from the group is the development of lumistine, a two-step process focusing on an alkylation and a nitrization, and using the DesiMath spec combined with flow, they were able to optimise temperature, time and the type of nitrization reagent in order to come to the most cost-effective manufacturing route for this now generic material. So if you want to learn more about that, there is a YouTube video that you can watch on that work and the reason behind it. So stepping away from screening now and going into pilot production, we've got a reactor of a couple of 100 mil here in a fume cupboard capable of producing at the multi-metric ton scale per year. So this is a process where we had a highly energetic piece of chemistry, typically highly diluted in batch, and the product is prone to polymerization if you don't control the heat release and the stoichiometry of the raw materials. So in this situation we used the infrared as a method of monitoring the complete consumption of the starting materials and the stable product quality. If we got any deviations, obviously with mass flow controllers we can modify the process on the fly in order to have that efficient, stable product quality. So here we're using a reaction time of 2 minutes, reaction temperature of 100 degrees for an exothermic process, and we're able to run solvent free. So by having that precise dosing of reagents, good heat transfer capabilities in the reactor, and a metal-free reactor to be resistant against the highly corrosive materials that are being used, we're able to generate a high purity product that doesn't need any isolation subsequently. So in this particular case, by optimizing the process well, we're able to increase product purity and significantly reduce isolation costs and raw material costs. Another benefit of PAT is the reduced operator exposure to raw materials and products that are generated in a production plant. So here Omnichem developed a multi-stage continuous flow process that not only increased the process safety due to exotherm control, but it also minimised operator exposure of a high potency product. So by being able to monitor what's happening in your process over multi-stages without having to physically take a sample, you have an operator benefit there and a safety profile increase. In addition, you end up with small units that you can test in the lab and then combine these units. You can see one highlighted in yellow. Combine these in order to get your production plant and move this into the production environment. So there's modular nature that allows you to qualify elements of your process before putting them together to deliver your multi-stage telescope process is a great advantage of flow, and also the combination of PAT in ensuring that this process can run unattended. So when you step out of the lab and you go into production, as I mentioned earlier, the type of process analytical tool that you require changes. You go from looking at detailed structural information to a situation where you know what molecule you're making and you understand the influence of critical process parameters on the performance of your process. So now you'll start looking at signals, not wanting them to change, and you're looking for stability and consistency. So this could mean you end up using pressure, temperature, or sometimes near infrared, for example, as an indication of your state of control. And this allows you to translate your process from the lab into manufacturing and retain all of the benefits that you had in your development cycle. And now you're simply looking for consistent product quality and a system health check. Is your reactor performing as it should? Are your pumps dosing as you expect? If you have the right temperature and if you have the right flow rates and you have the right hardware, then your product quality will remain consistent. So you go from a totally different reason for using PAT, but it remains just as valuable. And what you can see in this graph here is just the value absolute that you can get from a simple temperature measurement. So this is a piece of work published in NPRD recently by Merck, and thank you to them for showing what happens when a process is not going to plan. Here you can see that the temperature sensor has a drift on it, and it's steadily increasing the temperature in the reaction stream. This actually ended up being an indication of a fouling incident that was happening within the heat exchangers and the residence time units, and in order to generate the amount of material that was needed for a campaign, they operated this process with a cyclical washing, restabilization and material collection, and this allowed them over a 30-hour period to generate a couple of 100 kilos of the ethyl ester. Obviously though, it's not a stable process for long-term manufacturing, and the question you have to ask is, could they have identified this problem ahead of scaling up to a couple of 100 kilos? So they went back to the lab and they looked into the root cause, and they found that water ingress in the base was leading to decomposition. Insoluble KOH was being generated, and this was building up. So by screening solvents, adding THF as a suitable co-solvent, they were able to do an extended run for 2 hours. And what you can see on the right is an incredibly different picture to the original trip to the pilot plant. So what you can see is that previously fouling occurred after 10 minutes, and in the new setup an extended run of 2 hours was possible. So then you can transfer to the pilot plant with the security and the knowledge that you now have a stable process. So the important thing here is to realise that if you see something going wrong at the lab scale, it's only going to become more of a problem at a larger production scale. So I would advise running your small-scale reactors for extended periods of operation, looking for system instabilities and fouling. And once you've done that and you've had several hundred reactor volumes through, you can be sure that you've developed a stable, robust process. And at that point, look to scale up. So I hope through these few examples that you've been able to see the value of putting process analytical tooling into your continuous flow setups. And you can see the benefits that flow can bring to small molecule production. So it's not only the safety, but the speed of development and the increased control that you have when you're operating a continuous process, all the way through to reduced production costs when you look at your decrease in safety investment requirements versus your increased product quality. So if you're considering going flow, I would advise you define what you need to be viable in a project, identify together as a team what are the actual goals of the work, and then design your R&D, process R&D activities to target those. Work as a team. Get yourself a multidisciplinary team together. This could be chemists, chemical engineers, analytical chemists, QA, QC team members, in this way that you can prevent any barriers coming later down the line when you've developed a new way of working. And I would recommend partnering so that you can develop much faster. I'd like to thank you for your attention, and I'd be happy to answer any questions. If there's not time in the Q&A slot, please feel free to reach out by email or phone. I'd be happy to answer your questions at a later date. So thank you once again.